So here we're looking at, there's the ovary, okay, and here's the follicle from which a secondary oocyte has been released at ovulation. And we find that secondary oocyte sitting here in the uterine tube. So it had been released from the follicle in the ovary, and we now see it in the uterine tube. But recognize that it doesn't go directly from the ovary into the uterine tube. When it gets released from the ovary at ovulation, it's being released into the peritoneal cavity. And then it gets sucked into the uterine tube. There's an active process. The uterine tube has an epithelium that is ciliated. The cilia are beating, creating a current, and it's that current that sucks the secondary oocyte into the uterine tube. And it's normally here in this distal expanded portion of the uterine tube called the ampulla of the uterine tube. This is the region where fertilization normally occurs, and we can see those sperm approaching that secondary oocyte, and one of those sperm might fertilize it. But let's just pause for a second and think about something that might go wrong. If this secondary oocyte, when it gets ovulated into the peritoneal cavity, if it doesn't get sucked into the uterine tube, if it remains out here in the peritoneal cavity, what could happen? Well, these sperm that we see here, let's remind ourselves how those sperm got there. Right? Those sperm were deposited in the vagina, which would be over here someplace. The sperm went through the cervical canal, through the uterus, up the uterine tube, and that's how they got here. Well, when they get to this point, they don't suddenly stop swimming. They keep going. And so the sperm that we see in the uterine tube here, they're going to continue right on out the end of the uterine tube into the peritoneal cavity. So we need to recognize that a sexually active female will have sperm in her peritoneal cavity. Now, the sperm aren't able to live there very long. They live maybe 24 hours, maybe a little bit more than that, and then they'll die. But there will be sperm in the peritoneal cavity. And so if the secondary oocyte remains in the peritoneal cavity, that raises the possibility that fertilization could occur in the peritoneal cavity instead of occurring in the uterine tube where it normally does. If it occurs here in the peritoneal cavity, then development will proceed. And we'll see in a little while that when development reaches 6 days of development, implantation occurs. And that embryo will implant on day 6, no matter where it is. So if it's here in the peritoneal cavity, it can implant wherever, whatever surface it is in contact with. So it might be in contact with the ovary, it might be in contact with the uterus, it might be in contact with the rectum that is nearby. Whatever it is in contact with, it can implant into, and that would be called an ectopic abdominal pregnancy, an ectopic abdominal pregnancy. So the cause of an ectopic abdominal pregnancy is a failure of the secondary oocyte to get into the uterine tube, therefore it remains free in the peritoneal cavity, therefore fertilization can occur in the peritoneal cavity, therefore implantation can occur on a peritoneal surface lining that cavity. The major risk associated with that is that as a result of that implantation, a placenta will develop. Normally, that placenta develops on the wall of the uterus, where implantation normally occurs. And at the end of gestation, when that placenta separates normally during labor, remember the smooth muscle of the uterus is contracting during labor, and it's that smooth muscle contraction that squeezes down on all the blood vessels of the placenta so that the bleeding that occurs with placental separation is stopped by the contraction of the uterine smooth muscle. But if this placenta is not in the uterus, but is on some other organ that is not contracting in labor, then we don't have the mechanism for stopping the bleeding, and with separation of the placenta, the woman is at significant risk of exsanguination. Okay? And it's for that reason that an ectopic abdominal pregnancy puts the life of the mother at risk, and typically such a pregnancy would be terminated. Okay, so let's get back to what normally happens. Normally, the secondary oocyte does get sucked into the uterine tube, and normally fertilization does occur here in the uterine tube. Once fertilization occurs, remember the second meiotic division is completed, the second polar body is given off. We now have an ovum with an ovum nucleus and a sperm nucleus. Those two nuclei fuse together, and we now end up with a zygote. The zygote now has 46 chromosomes and is a diploid cell, 2N. That zygote will now start dividing. Now, this is no longer a gamete, so it's not going to divide by meiosis anymore. It's going to be dividing by mitosis. So the zygote will start to divide by mitotic divisions. So when it divides by mitosis, it gives rise to two cells, okay, just replicates itself, and that's called a two-cell embryo or two-cell blastula, okay. And then when those two cells divide by mitosis, that gives us 4 cells. When they divide by mitosis, it gives us 8 cells, and so on and so forth. Okay? But there's going to be something different about these mitotic divisions compared to other somatic cells in the body. When we look at other somatic cells, pick your favorite cell, an epithelial cell or a liver cell or whatever cell you like that's dividing by mitosis. When that cell divides, it gives us 2 cells that are half the size of the mother cell, and then those 2 cells grow. And when they grow to a certain size, then they divide again, giving us 2 cells half the size of those cells, and then they grow. And when they get to a certain size, they divide again, and so on. What's different here in these cell divisions is that you do not have cell growth in between cell divisions. So in other words, when this zygote divides into 2 cells, each cell is half the size of the original. It doesn't grow. And then it divides into 4 cells. So each of those cells is 1/4 the size of the original. When they divide, we end up with 8 cells. Each cell is 1/8 the size of the original, and so forth. So that the total cell mass of a 4-cell or 8-cell or 16-cell embryo is the same as a zygote. The total size is the same. We have more cells, but they are smaller cells. So this process continues during the first few days, and we go from 2 cells to 4 cells to 8 cells. That cell division, by the way, that occurs without any cell growth in between divisions is what's called cleavage division. Cleavage meaning cleaving meaning cut. You're just cutting the cytoplasm into smaller and smaller pieces. That's why it's called cleavage division. So that process occurs during the first several days of development. And we go from 2 cells to 4 cells to 8 cells to 16 cells and so forth. At some point, different people use different numbers. Most people say 32 cells. Some people say 64 cells. It doesn't really matter. At some point, we stop counting. We stop using numbers. And we just call it a ball of cells or a morula. So by day 4, we have a ball of cells, a morula, again, still the same size as the zygote. Okay? By day 5, something interesting happens. The cells continue to divide, but now they start to rearrange themselves. The way they rearrange themselves is that the extracellular fluid between these cells all starts to aggregate together into one large fluid-filled cavity, and the cells get densely packed on the other side here. And so we end up converting from a solid ball of cells, which is what the morula is, into a hollow ball of cells, hollow in the sense that we have this fluid-filled cavity here with an accumulation of cells on one side. And that is now called a blastocyst, And that's where we get to by day 5. Notice that we are still in the uterine tube at that stage. We're still in the uterine tube. Once we have a blastocyst, we can describe 2 different groups of cells in the blastocyst. Those cells that are forming the surface of the blastocyst, that can be called the outer cell mass, also called the trophoblast, and those cells that are clumped together on the inside, which is called the inner cell mass, also called the embryoblast. Now, when you see a word that ends in blast, That means it's a cell that will develop into something. You're probably familiar with words like fibroblast and myoblast and neuroblast and chondroblast and osteoblast. Each one of those is a cell that develops into a particular cell type. So embryoblast means something that will develop into an embryo. So the point is that it's only the inner cell mass of the blastocyst, only the inner cell mass that actually will become the embryo. That's a relatively small percentage of the total blastocyst. The outer cell mass, we said, is also called trophoblast. We know what blast means. Troph refers to nutrition. So the trophoblast will develop into something that's responsible for the nutrition of the embryo, which is, of course, the placenta. So the trophoblast will give us the placenta as well as some extra embryonic membrane. Okay? So that's what we have at day 5. A blastocyst with an embryoblast on the inside surrounded by a trophoblast. Those inner cell mass cells, those embryoblast cells, are the same cells that you'll often hear referred to as embryonic stem cells. In other words, each one of those cells has the capacity to differentiate into any cell type that will be found in the future embryo. So if you were to take a blastocyst, take the inner cell mass out of the blastocyst and dissociate those cells in the laboratory, each of those dissociated cells would be an embryonic stem cell. So the source of embryonic stem cells is the embryoblast of a blastocyst. Okay, so there we are on day 5. By day 6, that blastocyst has finally arrived in the uterus. And remember, on day 6, that's when implantation typically occurs. So since it's typically in the uterus on day 6, implantation occurs into the endometrium of the uterus. But that also means that if for some reason the pathway down the uterine tube to the uterus was in some fashion obstructed so that the migration down the uterine tube was slowed down, so that by day 6, instead of getting to the uterus, let's say it was still here or here or here, well, that means that on day 6 it would implant wherever it is. So if it's in the uterine tube, it would implant into the wall of the uterine tube. And that would then be called an ectopic tubal pregnancy, an ectopic tubal pregnancy. So the cause of an ectopic tubal pregnancy is an impairment of the migration down the uterine tube so that by day 6 it hasn't completed its migration into the uterus, it's still somewhere in the tube. Often what's causing that is scarring of the uterine tube, which typically would be a result of an earlier pelvic inflammatory process. So women who have a history of pelvic inflammatory disease are at significantly higher risk of having an ectopic tubal pregnancy because of the possibility of scarring of the uterine tube, which would impair the migration of this developing embryo down the uterine tube. But again, normally it does get to the uterus by day 6, so implantation does occur into the endometrial wall of the uterus. So let's see what happens in implantation. So this diagram is an enlargement of that. And what we see is the blastocyst. There's the inner cell mass that'll become the embryo. Now we see that the trophoblast is starting to develop these finger-like projections of trophoblastic cells. Those are called villi, trophoblastic villi. And we see that those villi are eroding their way into the endometrium. And that's what implantation is, an erosion into the endometrium. Now, where are those villi coming from? What we're getting is a tremendous elaboration of trophoblastic cells, specifically a kind of trophoblastic cell called syncytiotrophoblastic. So, what does that mean? Well, the trophoblast is made up of two kinds of trophoblastic cells, cytotrophoblast and syncytiotrophoblastic. Cytotrophoblast, cyto means cells, so cytotrophoblast is the trophoblast that's made up of individual cells. And what cytotrophoblast cells do is really just one thing. They divide by mitosis. They divide and divide and divide. They are very mitotically active cells, doing a lot of cell division. The daughter cells of those cell divisions will do one of two things. Either they will be just like the mother cell, they will remain cytotrophoblastic cells, which means they will then go on and divide, because that's what cytotrophoblast cells do. Or the alternative is the daughter cells can fuse with one another, and as a result of that fusion, we now have a multinucleated cell, and we call that syncytiotrophoblasts. So it forms a syncytium, a multinucleated cell. So those fused cells are now forming the syncytiotrophoblasts. But remember that syncytiotrophoblasts, or any syncytiotrophoblasts do not divide. There is no mitotic activity any longer. So syncytiotrophoblastic cells are not able to divide. What we're seeing here is a tremendous growth of syncytiotrophoblasts. So how are we getting more and more syncytiotrophoblasts if they are not able to divide? And the answer to that is that the cytotrophoblast is what's continually dividing, and many of those daughter cells are fusing to give us more and more and more syncytiotrophoblasts. So the source of the increase in syncytiotrophoblasts is the fusion of daughter cells of cytotrophoblasts. But keep in mind that you can't have all the cytotrophoblastic cells fusing, because if that happened, there would be no more mitotic division and there'd be no more source of more cells. So some of the cytotrophoblast cells have to be kept in reserve and kept as cytotrophoblast so they keep on dividing, while the rest can then go on and fuse to give us more and more syncytiotrophoblasts. And it's the syncytiotrophoblasts that we see invading into the endometrium. Now, another important role for the syncytiotrophoblastic cells is that they secrete a hormone called chorionic gonadotropin. Chorionic gonadotropin is a hormone that will go back and signal the mother's ovary to tell the ovary to retain the corpus luteum that forms after ovulation. The corpus luteum is what's producing progestins, and the progestins are necessary to maintain the endometrium in order to maintain the pregnancy. So the chorionic gonadotropin is basically telling the mother that she is pregnant and that she needs to continue to produce progestins from the corpus luteum in order to maintain the endometrium. So it is basically the signal for pregnancy. It is the marker for pregnancy. So pregnancy testing that is done clinically is an assay for chorionic gonadotropin. So as we develop syncytiotrophoblasts, we're producing chorionic gonadotropin. When the levels of chorionic gonadotropin get high enough to be detectable, we then get a positive pregnancy test, which is, to be very precise, is not really saying that there's an embryo there. To be more precise, it's saying a placenta is starting to develop, because that's what the syncytiotrophoblast is going to become, a placenta. Okay, so that's what happens by the end of the first week.